Hello and welcome to my presentation. I am Martin and I will be introducing you our methods for deciphering the secondary metabolism of the human gut microbiome. The impact of the microbiome on the health of the human host has been shown in countless cases. However, most of them, the exact mechanisms still remain unknown. One of the ways the microbiome may be interacting with the human host is through the production of secondary metabolites. These secondary metabolites are typically being produced by entire enzymatic pathways encoded in colocalized genes inside regions called biosynthetic gene clusters. These secondary metabolites can then have an impact on the microbiome itself, or also on the human host by regulating the development of pathogenic bacteria, or by directly getting involved inside certain mechanisms of the human host health, like carcinogenesis. To discover BGCs from genomic and metagenomic data, there are two families of methods. The first one is called rule-based BGC prediction, which uses a PHMM library to detect signature genes that are specific to a BGC class, or at least a family of BGCs. The input sequences are annotated only to look for these signature genes, and then some rules are applied to remove false positives. This approach has extremely high precision in the sense that it leads to the discovery of BGCs that contain these signature genes, but it cannot find BGCs with novel architectures. The second type of approach, however, can discover new architectures. It uses domain annotation of all proteins as input features to a sequence learning model. And then the sequence learning model can output BGC properties across the whole genome. However, the existing methods that are model-based were clearly not satisfying in terms of accuracy, with a vast number of false positives being produced by both cluster finder and DBGC. To address this problem, we developed GECKO, which is gene clusters prediction with conditional random field, our new method for using a model-based BGC prediction. To do so, we start by predicting open reading frames within prokaryotic genomes and metagenomes using Prodigal. Then we annotate these proteins using protein domains from PFAM to create domain vectors. These domain vectors are then used by our conditional random field to compute probabilities for BGC regions. When we evaluated this method against the other model-based methods like DBGC and cluster finder, we found that GECKO massively outperformed them. You can have more information in our preprint from 2021 or from the GECKO website. Now that we developed a new method for BGC discovery with satisfying levels of accuracy, we then went on the annotation of the biosynthetic landscape of the human gut. To do so, we assembled two collections of genomes, roughly 50,000 genomes from the progenomes database that were found from species with high prevalence in the gut, and almost 300,000 metagenomes, assembled genomes from the UHTG catalog, which originates from human gut isolated metagenomes. We then annotated our genome collection with both anti-SMASH and GECKO, where we found roughly 800,000 BGCs. These BGCs were then clustered at the nucleotide level through different steps of redundancy removal in order to obtain 45,000 gene cluster families. To visualize these gene cluster families, we extracted the representative cluster of each family and then represented it with its PFAM domain composition before plotting it. The resulting high dimensionality vector representation can be reduced into a UMAP, which then shows the different regions of the biosynthetic landscape. In terms of key results, we showed that roughly 2/3 of the gene cluster families are confined to a single species, that the human gut shows a higher diversity of ribosomal peptides compared to other habitats that are like soil or marine environments, and that also GECKO finds the highest number of novel clusters in Bacteroides and Parabacteroides, where we selected candidate clusters for validation. However, despite all these predictions, it's still hard to resolve the functionality of these gene clusters. more than 96% of the JIT cluster families contain no experimentally validated member, and apart from the cluster type, not much is known about the prediction. Now, if you look at the compounds being produced by bioscientific gene cluster, they tend to vary in functionality, in bioactivity, and in structure. However, you may be able to see some common patterns in some of them, like for instance, the presence of these thiazole cycles. And actually, you can classify these compounds even more using some chemical taxonomies, such as the one provided by Classifier, where given a molecule, you obtain an entire taxonomical classification of this molecule. Now, our question was, in order to resolve all our unknown BGCs, can we actually try to predict the chemical taxonomy for a compound being produced by a BGC entirely from its genomic region? So to approach this problem, we developed a novel method called CONC, which stands for Classification of Natural Product Chemical Hierarchy, where we yet again start by predicting the genes and the protein domains from each BGC, but then using independent logistic regressor classifiers to predict chemical classes for a given BGC. The result is actually a vector of predicted classes that can be inspected for chemical features of interest. For instance, if you look at the predictors for different biocycles that appear in biological molecules, we can see that our predictors is able to predict the presence of a thiazole or a beta-lactam cycle very accurately, just based on the domain composition of a BGC. This can actually be used to go back to the human gut, and instead of looking at a very coarse-grained annotation of types, we can now label with a much more fine-grained annotation our human gut landscape, where then we can see that, for instance, there is a concentration of thiazole in BGCs that most likely belong to thiopeptide class on the southwest of the landscape, whereas beta-lactam cycles, for instance, are much more scattered across the entire UMAP. Another interesting result of this approach is that the predicted classes that we obtain can actually be viewed as fingerprints, and these fingerprints can be used to search a catalog of compounds for similar classes to a prediction, given that this catalog has been predicted in the same manner. Luckily, using Classifier, we have access to classifier annotations for the entirety of the natural product atlas. So we can, for instance, take a BGC. For this example, I'm taking a known BGC, which is the atrobimycin BGC from Streptomyces atrovirens. It's not a gut bacterium. Then we can pass it through CONC, where it will produce the entire chemical taxonomy for this BGC based on its composition. Then we can look for the natural product atlas for a similar compound. And this is the compound that it found based on this fingerprint. And given that this is a known BGC, we can actually see the true compound being produced by the BGC from the literature. And we can see that despite not being the same compound, the prediction made by CONC is actually quite close in the sense that it shows a lot of structural similarities, like the presence of this cinnamic acid moiety, or just the fact that the molecule itself is a macrolactam. This is actually made possible by the fact that this BGC is quite similar to a BGC from MyBig, but shows a lot of internal sequence variability, and thus a pairwise alignment may not reveal the same kind of features that CONC is able to capture. Finally, one of the key interesting results of using a logistic regressor for this task is that we can train it with a lasso, which clearly increases the sparsity of the learned weights for each regressor. It means that for each class, we can now extract which weight contributed to the classification and build a network. So in this network, I extracted for each class the two most contributing domains as learned by the logistic regression model and basically link them. And what's interesting with this kind of network analysis is that you can then inspect that the training has basically went on properly, And for instance, you can see that for certain domains like the tryptophan halogenase, Conk has learned that the presence of the tryptophan halogenase is associated with the production of a compound that is either a halobenzene or an organohalogen compound, which makes sense. So it can be used to validate what the model is learning. But then we can also use it as a discovery approach, since this is data-driven, for potentially proposing new functions for domains of unknown function. So for instance, in this domain of unknown function from PFAM, we can see that the model has learned that it was linked to the production of thiols or imidothiazic acid. So it may be a clue for its actual function. With this method, we hope to improve the knowledge of existing biosynthesis, as well as improving the resolution of biosynthetic gene cluster inside the gut. And with that, I would like to thank you and thank all the people that work on this project. You can find more on my personal, on our group Twitter. And also, please feel free to come if you have questions at any point during or after the conference.